Thank you so much. Thank you so much, Yasser, for the kind introduction. Yeah, and I want to thank you for the opportunity to be here today and to be presenting our work. Yeah, so as you could hear from the kind of the brief bio, my training has always been in reproductive biology, and pretty much in understanding how embryonic development happens from the embryonic standpoint, but also from what comes just before embryos, which are germ cells. And so in my lab now, the theme of my lab is really to kind of build on that knowledge and try to still understand embryonic development and reproductive biology, in particular in the context of human development, because of course embryonic human development cannot be easily studied because of the fact that, of course, there are ethical and technical implications and complications. So I want to study the biology of that process, but at the same time, I want to really hijack in a way or leverage those principles and apply those principles to tissues that are not embryonic and not germ cells, and utilize them for the long-term goal of really trying to tackle the problem of aging and aging-associated disorders or indications, in particular in the context of women's longevity and women's health span. So I will give you a very quick overview of a couple of programs, a couple of research programs that are ongoing in my lab. I'm happy to take your questions, even during the talk, if you wish. So as I said, at a very broad level, what my lab does is, on the one hand, to really try to study, model, and understand human pre-implantation and early post-implantation developments, for the sake, again, of developing, for example, disease modelings, or for the sake of understanding the biology of these complex processes, and utilize, and develop an in vitro model to really study those processes. But at the same time, to utilize those principles and apply them in the context of women's health and women's longevity. So why? Well, I think that the aging program, the aging problem, generally speaking, is a huge problem. So we all know that growing older or getting older puts us at risk of developing one or more of these chronic conditions that you see on the left side of the slide. So people that are 65 years or older have an 80% chance of developing at least one of these conditions. And about 70% of them are at risk of developing two or more. Why? Because they get older, because they age. So aging is the #1 risk factor for all of these diseases. Now, if you combine that with the fact that in about 30 years from now, there's going to be approximately 20% of people that are going to be age 60, 65 or older, that means that in 30 years from now, 2 billion people are going to be at risk of developing one or more of these conditions. And so I personally think that this is really kind of a huge problem, and this is something that is going to impact our society dramatically, and I think it's about time to do something about that. Now, if you zoom in even further, there is obviously within this general problem or situation, there is also a women-specific aging phenotype in a way that is severely understudied and needs to be addressed. And so, well, we all know that, of course, there is the aging of the germ cells, for example, in women is something that is an obvious issue. Not just because the women then at some point in life they are no longer capable or reproductively kind of capable, but that also has a huge impact on their health span. So here's just a few facts. For example, the timing of menopause onset is linked to the susceptibility to age-related morbidity and mortality. So the sooner a woman enters menopause, the shorter the lifespan, and the higher the chances of developing age-related comorbidities, with time. And it's also, the timing of menopause is also inversely correlated with the lifespan and health span. And there is an interesting study done in, I think, approximately 10 years ago, showing that for every 1 year increase in the menopause age, the age-adjusted mortality rate was decreased by 2%. Now, you take that and you combine also with data coming from animal models, and you really start seeing like kind of, a pattern here, and you start seeing, understanding, what I mean. So for example, in mice, if you do ovarectomy, so if you just, take away the ovaries, which is in a way, a model of early menopause entrance, this leads to shortening of lifespan and reduced health span, affecting a variety of different organs, systems, and tissues. On the flip side, if you take older mice, you ovarectomize them, so you remove the ovary and you transplant young ovaries, you have an increase in lifespan and a decrease in morbidity and increase, health span in these mice. So this really tells us that, again, there is a global aging, quote unquote, pandemic going to happen in the future. And, this is a very important issue, in particular when it comes to women's health and women's longevity. So the goal of my lab really is try to start addressing this problem and start developing new potential therapies to tackle this important problem. And this is kind of in line with what's the field in a way is kind of moving towards. So up to now, so if you imagine that this is kind of the normal trajectory of lifespan, up to now, medicine has developed prevention kind of intervention to kind of delay that process, right, from happening. But by doing so, of course, we live longer. But at the same time, we extend the time window by which we are more frail, and we are at risk of developing one of those comorbidities that I've shown before. So now there is a kind of a new radical way of looking at this problem. So what about intervene? What about developing intervention strategies that could really try to tackle this problem at its very root, right? And for example, not just preventing, but stalling the process of tissue aging, or to some extent even reversing it, so that a particular aging-associated disease, doesn't happen in the first place. And so, again, the research in my lab kind of focuses on this big problem and on developing new intervention strategies. How? Well, I'm going to share with you today, very briefly, two potential approaches. So one is ERA, which is the idea of epigenetic reprogramming of aging, which was in a way informed by the work that I previously did in the context of embryonic stem cell and induced pluripotent stem cell reprogramming. So the idea very simply is about transiently reprogramming the cells using a cocktail of mRNAs, so that now you turn an old dysfunctional cell into a young, functional, juvenile cell that can, behave much better, in the body. And the second approach is kind of tackling a little bit more the problem of reproductive aging in women, developing stem cell-derived ovaries that could potentially be used not for the purpose of reproduction, but for the purpose of extending, similar to what has been shown in mice, extending or impacting the health span of postmenopausal women. So I'm going to start with this. So again, the idea comes again from the fact that I have been studied embryonic development and germ cell biology for a very long time. So if we think about embryonic development, in particular pre-implantation embryonic developments, and if we think about gametogenesis, what's really striking about these two phenomena is that both of them are characterized by a massive epigenetic resetting of the genome, characterized, for example, by massive DNA demethylation, histone modification changes, histone positioning in the chromatin. So there are two waves of epigenetic reprogramming, one of them happening the first couple of days of embryonic development, and the second one happening in the germ cells as they form and mature in the organism, that are characterized again by massive DNA demethylation and massive epigenetic resetting. Now, these two phenomena, of course, have a function, and the biological function is really to turn one set of program, for example, the germ cell program, into a new set of program, the embryonic program, during the pre-implantation development, or in the case of PGCs, to really set up a gender-specific set of programs that separates, for example, male cells, the sperm, from the female cells, the eggs. But I think that there is a lot of biology here that can be learned and studied and applied to cells that are non-embryonic and non-germ cells, for the purpose, again, of resetting the epigenetic clock and resetting it to a more youthful state. So how do we do this? Well, fundamentally, what we use as a model, sorry, I need to move, okay. Fundamentally, what we use as a model, as in a platform, is the idea that was, or the technology that was developed by Shinya Yamanaka in 2006, 2007, which is very simple and was groundbreaking. The idea is that you can take any somatic cell, so any cell of the body, and by the addition of just a handful of transcription factors, for example, OCT4, SOX2, KLF4, and CMYC, you can reprogram the cells. So say, for example, a skin cell, you can reprogram that cell to an embryonic-like cell. They're called iPSCs, which stands for induced pluripotent stem cells. So you can take a skin cell again, for example, and you can turn that skin cell into an embryonic-like cell. Now, that embryonic-like cell has the potential to be redifferentiated, not only in the skin cell you started with, but also in a variety of different cells, for example, heart, liver, blood, brain cells, and so on and so forth. Now, obviously, this is great because now you can potentially use these iPSCs to generate tissues, organs that are immunocompatible, right, with the donor. But what's really also interesting, and that was for a long time kind of understudied or not fully understood or appreciated, I have to say, is the fact that during this process of reprogramming, there is a change in cell identity happening. So again, you turn a skin cell into an embryonic cell. But what's also happening is that you're making an old cell into a very youthful, fully functional cell that now is juvenile by all means, has longer telomeres, can deal with DNA damage like a very young cell, has very functional mitochondria, and so on and so forth. And what's also really intriguing is that now if you take this embryonic cell, which has been rejuvenated, and you make, let's say, another skin cell out of it, the skin cell that you're making is not old as the skin cell you started with. It's actually very youthful. So the idea that we had in the lab was, well, we know that these two phenomena are happening here. So you have a change in cell identity and a change in age, in cellular age. What if you could decouple these two processes? What if you could make a cell younger without changing its cell identity? Because if that was true, then you could really think about a completely new concept in regenerative medicine. So you wouldn't have to go to an embryonic-like state and then redifferentiate the cells into, for example, liver cells or brain cells, but you would just go into the tissue, reprogram it partially, and just make a younger version of that tissue, in situ. So that's kind of the general idea. And so we tested this hypothesis, and we thought that maybe this could be a function of time. So the degree of cellular rejuvenation, which is here defined as Y, could be a function of how long the reprogramming occurs. So how long the reprogramming events are taking place, in the cells. And we published this work in Nature Communications a couple of years ago. and this is really kind of the foundation of the science in my lab, and the foundation I have also to say, you know, as a disclosure, of the company, of the startup that I have co-founded. So the two questions, of course, back then were, well, how do we make sure to precisely vary X? Because again, if it's a function of time, we have to really make sure that we can very finely control the timing and the duration of the reprogramming. And after that, how do you measure Y? So what is the degree of rejuvenation that you are seeing by working at the nuclear level, at the epigenetic level in the cells? So to control X, which is, you know, the duration, what we chose was to use mRNAs. And I have to say that this was kind of a gamble back then. So we started this project at the end of 2014. But I have to say that, you know, this paid off big time. And kind of sadly also, thanks to what happened with COVID, we're really understanding now how the power that mRNAs have when it comes to gene therapy and regenerative medicine. And so the advantage that mRNAs give is that you can really very easily kind of orthogonally control all of these variables that you see up here. From the number of factors that you're using, the types of factors that you're using, their sequence, their stability, their cytotoxicity, the relative ratio of the different factors that you're using, obviously the duration and the amounts, but most importantly, and again, this is something that we have learned very recently, most importantly, mRNAs give the opportunity to really scale up the production and really significantly reduce the cost of the drug product. And I particularly like this because, you know, it's a way to make this, to democratize in a way the system and make it affordable, not just to wealthy people, but, you know, pretty much to everybody. And so really what's really left to the cells is the translation, like the proteins that are then generated from those mRNAs. And again, in a very short, or in a nutshell, what we demonstrated in the paper was that you can take an old dysfunctional cell, and by providing for a given time a cocktail of reprogramming factors through mRNAs, you can make that cell into a very youthful cell that is rejuvenated across most, if not all, of the hallmarks, of the canonical hallmarks of aging. We have now demonstrated this, you know, across many different cell types, human, all derived from naturally aged individuals, because again, aging is a very complex phenomenon, and I think it needs to be studied in its complexity. And so I want to very briefly walk you through the data and think about the potential implications of this. So here, for example, you see that we took young and aged fibroblasts on the left, or young and aged endothelial cells on the right. So you see that for every marker that we looked at, and we tried to be as comprehensive and holistic as possible, for every marker that we saw or we looked at, we see that with time, so comparing blue, young, and red, old, you see that, you know, there is a change happening for that particular marker, right? When we transiently expressed a set of factors, we saw in the aged cells, we saw that actually that particular, marker changes, and it changes in a youthful direction. And so this, for example, is true for HPA-1 gamma, which is an heterochromatin-associated protein. It's true for fibroblasts, it's true for endothelial cells. But then you start looking at all of the other markers, and you see that the same trend, is true for other markers too. And every time, for example, a marker gets higher with age, you see that there is an opposite trend. So you see a decline with the treatment. So, for example, for mitochondrial reactive oxygen species. The next thing that we did was, well, maybe this is due to some sort of artificial or artifacts due to the fact that the cells are stressed. And so what we did was to do the similar experiment, but this time instead of transfecting the reprogramming factors, we transfected GFP mRNA, so an RNA that encodes for a fluorescent protein that it's not supposed to do anything to the cells. And what it was really interesting to see that, for all of the markers that we saw or we looked at, we either did not see any change in the expression of that specific marker, or if we did, we saw a change in the opposite direction. So the cells were actually kind of behaving even worse or older than the untreated cells. So this was really suggesting and telling us that something special about the transient expression of the reprogramming factors was really providing this multifaceted rejuvenation profile that we saw in the in vitro. Then we looked more holistically and comprehensively at other hallmarks of aging. So we looked at the transcriptome, for example, so the genes that the cells expressed. And again, you see here that if you take young cells and old cells, fibroblasts in this case, you see that there is a gene expression profile difference. So some genes get turned off with time, and some genes get turned on with time. When we treated the cells, so we took the aged cells and we treated them with our transient reprogramming, we saw that for the same set of genes, we could reestablish a profile of expression that was more likely that observed in young cells. Then we looked at methylation, so DNA methylation, in collaboration with Steve Horvath, who developed the methylation clock at UCLA. And we saw that also at the methylation level, there is a decline in methylation age. So the methylation profile of the cells seems to be more closer to the methylation profile that you have in younger cells. So what are the potential clinical implications of this? Well, this is very new data that we just got. So, for example, you can start thinking about using this technology, for example, for improving wound healing in aged skin. So here you see again very preliminary data that I'm happy to share with you. So in this case, we took a 16-month-old mouse. Okay, this mouse is a transgenic mouse. So it expresses 4 of the 6 reprogramming factors that we used in the work that I've shown you before, and it does so by adding doxycycline, which is an antibiotic, to the media. So we took the skin of this mouse, old mouse. We made skin biopsies, and we put the skin biopsies in culture in a trans well. Okay. After doing so, we also generated a burn wound into the skin, circular, as you can see. And then we divided the samples into 2, into 2 categories. One untreated, and one treated with a drop of doxycycline added here to the side of the wound. Okay, so again, keep in mind, doxycycline goes into the cells and it turns on the 4 factors that we want to be expressed in the cells. Okay, and it's really intriguing because you can do histology, you can do all sorts of assessments here. So for example, you can take the skin and you can do histology, and for example, you can measure the width of the wound. So you measure basically the distance between, so this is the burn area, as you can see, so there is no epidermal layer here. And you can start measuring, for example, the distance between this boundary here and this boundary here, so which is basically the intact tissue which is surrounding the wound. Okay, so here you see, for example, after 4 days of treatment with doxycycline, you see that there seems to be a trend. So the treated sample seems to start shrinking the wounded area at a faster rate or a faster pace than the untreated sample. And if you go for up for 4 days more, you see that there is actually a dramatic decrease in the wounded area. And again, this suggests that the healing capacity of the old burnt skin in the presence of the 4 reprogramming factors expressed just for 8 days seems to be much enhanced.
Vittorio.
Yes.
Quick question. Have you been able to do this with a non-transgenic mouse to show that the doxycycline doesn't benefit the wound healing process just as by being an antibiotic?
Good question, Virginia. Not yet, but that's going to be our next control. Yes.
Okay.
And what is the possible explanation of this? Well, in part, I think we believe that, for example, there is an increased proliferation at the boundary sites that is induced by doxycycline, and so again, potentially by the expression of the factors. So you see here that at the borders here, there is a massive proliferation of keratinocytes that starts to grow and then kind of migrates and close the wounded area. So again, this is very preliminary. There is still a lot of work that needs to be done in making sure that we really standardize, for example, the size of the burn, the temperature that we use for the burn, the depth of the bone. But it's very encouraging data that building up on what we have shown before, really potentially could be explored as an intervention, for example, in the context of wound healing, just to make an example. So this is all exciting, but I think that what's really even more exciting is the fact that potentially you could even start thinking about using ERA, so this idea of rejuvenation, and apply it to stem cells. So, well, the work that I've shown you before is done on fully differentiated cells, fibroblasts, keratinocytes, and so on. But if we could potentially rejuvenate the stem cells, so the cells that in every tissue are responsible for the full regeneration of an organ or the tissue, you could potentially have a very long kind of, effect, right? Because you're really rejuvenating the cell that is responsible in the long term for the regeneration of the whole tissue. And so basically what we did here, and I'm going back again to published data, was to really test this idea and do this in muscle stem cells. So very quickly, what we did here, we did the experiment in mice to begin with. What we did was to isolate the fibers, isolate the stem cells from the fibers, and this was done in collaboration with Tom Rando's group, and Marco, a good friend of mine, had developed a way to really isolate and keep the stem cells quiescent and fully and preserve their potency in vitro. So we separated the cells into 2 groups, the untreated and the treated. And the treated were treated only for 2 days. So only 2 pulses of mRNAs for 48 hours, and that's it. And then the cells were immediately transplanted back into a host, the immunocompromised mouse. And at the time of transplantation, the muscle was also injured by a cardiotoxin, barium chloride injection, to basically destroy the tissue and challenge the cells to repopulate and regenerate the entire muscle. In some cases, we also did a second injury 2 months after the transplantation to really also measure if these could have a long-term effect on the regenerative capacity of the cells. The treated cells, in both cases, the cells were also tagged with luciferase and GFP, and the untreated cells were transplanted in the left leg, and the treated ones were transplanted on the right leg contralaterally to also make sure that the cells were in the same systemic environment in a way that could obviously have an impact. And you can see here that, the aged untreated cells, so this is the bioluminescence, so this is the light that is coming from the luciferase expressed by the stem cells. And it kind of gives you an idea of the regenerative kinetic of the process. So you see that over the course of 11 days, this is how the untreated aged stem cells behave and grow. You compare that with the blue one, which is the untreated young stem cells, and there is obviously a massive difference in the regenerative capacity. But now, if you take the cyan, which are the aged treated cells, you see that the age treated cells behave like young, if not, they behave as good or if not better than the young untreated cells. When you do morphometric analysis, you see that there is no sign of tumorigenesis, because of course the caveat could be that the cells have lost their identity, right? And now they're making tumors, for example. But that was not the case. We followed the mice for up to 6 months, and there was no sign of tumorigenesis whatsoever. There was only more fibers and thicker fibers, as we quantified down here in these two graphs. And then when we did the experiment, when we did a second injury 2 months after, we saw that there was a memory of that rejuvenation process. And so again, the treated age cells were behaving like young, untreated cells, suggesting that something was really reset at the epigenetic level in the cells. And the cells now were really behaving like young cells, even for after a long period of time, which is 2 months, which is in the lifespan of the mouse is a pretty significant amount of time. Then we did similar experiments in, this time we did an endpoint analysis at 30 days. We transplanted the cells not in young mice, but in aged, in age-matched old mice, because the idea here was to kind of put the cells back into their quote-unquote nasty aged environment they came from, right? And what we did here was to measure after 30 days the strength of the muscle that received the transplantation. The strength was measured in vitro. It's called tetanic force. So you basically you measure the contractility of that muscle, and you can see here that if you compare the average force of the tibialis anterior of an aged mouse, that's what typically you get. And it's obviously much lower than the average strength that a young untreated mouse muscle exerts. When you transplant the aged muscle with aged stem cells that have not been treated, you restore the physiological function, force of that muscle. And if you do the same experiment, but you transplant now aged treated cells, you restore the physiological force of a young untreated muscle. So it's about 40% of muscle induction, which is pretty significant, after 1 month. And lastly, we did the same experiments with human stem cells, and we obtained very similar results. What was really interesting also here was that we could stagger the samples by age groups, and we saw that most of the significant effect was seen in the samples that were coming from the age 60 to 80 group, suggesting again that there was a process or a phenomenon of aging here that could be restored and reset by the treatment. So, well, I'm gonna go very quickly here, but basically the way we are thinking about this is the following. So if you think about embryonic development and you plot the functionality of the cells versus the age, you start from embryonic cells, which are very young, but non-functional because they haven't yet differentiated. And with time, what happens is that the cells reach an older stage and a much more functional stage. With time, obviously age increases and functionality decreases, because we know that with aging, cells lose their ability to function properly. Classical interventions, for example, I don't know, caloric restriction, rapamycin, metformin, and things like those, can only partially rejuvenate the cells, and bring them back in time. What ERA does is really kind of significantly moving the marble back into a young but still very functional stage or state, thanks to the process of epigenetic reprogramming. So we are obviously looking at many different potential implications. So starting from kind of starting, for example, from skin aging, but also looking at other problems like sarcopenia, neurodegeneration, age bone fractures, and so on and so forth. And I have to say that I'm actually very pleased to really see that now the field is booming. And there's many different labs now that are really confirming what we have seen in many different contexts. So for example, there was a science paper published just a couple of months back showing that the same idea can potentially be utilized in cardiomyocytes, post-infarcted cardiomyocytes. So the cardiomyocytes have lost, not because of aging, but they have lost because of development, their ability to regenerate, right, and to grow. So transient reprogramming has been shown to be reverting those cells to an embryonic-like cell, to an embryonic-like state, where the cells are still capable of proliferating and regenerating the infarcted heart. And so, this really tells you that this idea potentially can be applied to many different contexts, and we are testing it, in many different contexts. For example, neurodegeneration. Okay, so that kind of concludes the first part of my talk, and very briefly, I want to go into the second one. I don't think I will have the time to go through everything, but and it would be the idea now to utilize stem cells to generate ovaries that could potentially be used to kind of restore a more juvenile reproductive capacity in the woman, not for the purpose of having babies, but just for the purpose of feeling better and having an increased health span. How do we want to do it? Well, we want to do it kind of, going back to the original idea. We want to actually utilize induced pluripotent stem cells to generate germ cells and to generate somatic cells that together could be used to form a reconstructed ovary that could be, again, potentially transplanted into postmenopausal women. So where are we with that project? Well, we are at a very early but exciting stage. So I'm sharing with you some data that are currently in a paper that's under revision. Well, you know that when it comes to, for example, reconstituting an ovary, you have two challenges there. You have to, on the one hand, the challenge of generating the germ cells, but on the other hand, you also have the challenge of generating the somatic cells, the granulosa cells that go together with, or that complex with the germ cells to form follicles. So we have figured out a way to efficiently generate germ cells. Very briefly, what we have done was really to develop a monolayer culture, so a 2D culture that can, in as short as 3.5 days, generate millions of germ cells. And when we compare the germ cells, those in vitro generated germ cells, with the fetal germ cells that were studied by a Chinese group, we can see that the germ cells that we are generating in vitro are transcriptionally very similar to the mitotically active proliferative germ cells that you see in the gonads. So we think that we are generating bona fide germ cells that are kind of the equivalent of the early post-migratory germ cells that colonize the fetal gonads. And so our next goal now will be to use these germ cells and to combine them either with fetal granulosa cells that I'm very thankful and grateful I'm getting from Virginia, or inducing those fetal-like germs, granulosa cells from embryonic cells or from iPSCs using a set of transcription factors. And this is an exciting collaboration that we are setting up with Roger. To basically kind of mimic and recapitulate what happens during embryonic and fetal development, which is, again, the formation of these follicle-like structures, which derive from the interaction of a PGC of a germ cells and the interaction with somatic cells that kind of surround the germ cells and help the germ cell to grow. So to go back to the original goal, I think that we can really start thinking about the aging problem and in particular the women's longevity and health span in a completely different way. We can start thinking about rejuvenating the tissues using, for example, ERA, but we can also start thinking about a more systemic approach and, for example, rejuvenating the ovary, and by doing so, pushing and promoting a more healthy and juvenile, for example, hormonal kind of phenotype and that potentially could have implication again for health span and lifespan in postmenopausal women. And so with that, I just want to thank the people in the lab, the collaborators, the department, of course, Leslie, Virginia in particular. My lab is here in the SIM1, the funding, and again, the people in my lab. I mean, it's a very small lab, but it's a very productive and full of talented people. And I want to thank you all for your attention. I hope I ran 1 minute over. Sorry.
Vittorio, thank you so much. This is just wonderful and a little bit surreal and a lot to process. The potential just seems amazing. I'd like to open the floor to questions for a couple of minutes.
Yes, sir. I have one for Vittorio. Really fascinating. In your mouse model of wound healing, I wondered whether you considered another kind of injury, namely a surgical incision, as opposed to the burn. This, of course, would have implications for the millions of people undergoing surgical incisions or having lacerations and other kind of injury. And you could certainly measure it the same way, I think.
Yeah, absolutely. Yes, we are now expanding this and extending this to other wound models. Absolutely. So yeah, and I have to tell you the truth that right now we are using these mice just because they're experimentally, easier to use. But of course, if we see any proof of concept, the idea would be to start working with human tissue and start delivering in vitro or ex vivo locally the same cocktail, the mRNAs that we have used for our in vitro studies, and start developing some potential clinically friendly, interventions. Yeah.
Right.
Yeah, along the
Vittorio, following up on the human applications, I wondered what thoughts you have about ensuring the safety, because in a human application, they have a much longer duration of the potential for dedifferentiation or tumorigenesis.
Yeah, very good point.
Advice.
Very good point. I mean, so far our data suggests that if you really make sure, so there is a number of quality of QCs that you have to put in place to make sure that absolutely that the identity hasn't been lost. It seems to be a pretty remarkably fast process, and again, you could think about, doing this treatment for just a very short time, making sure that the cells haven't lost their identity, but still get a significant, outcome from that. So I don't want to be over optimistic, but I think that, there's definitely room for improvement. And the good thing about the mRNAs is that you can really, you have a lot of leverages there to control the safety aspect, because you can, for example, design some regulatory sequences on the mRNAs, and for example, by virtue of those regulatory sequences, have a shorter or an extended lifespan of the mRNA, and so have a kind of a shorter or a longer, potential efficacy time window.
Vittorio, I'm going to take the privilege of the last question here and just ask a very pedestrian question.
Sure.
As you rejuvenate some organs and you rejuvenate some cells, what does that mean unless you rejuvenate all organs and all cells? You end up with strong muscles and an aging heart, strong bones and aging muscle.
Very good question. So it's a complicated question. I think so. There's two different ways of thinking about that problem, Yasser. So one problem is like, so I want to make an impact. I want to make a difference when it comes to quality of life of people. So if I have to live for 20 years or 30 or 40 years with osteoarthritis, I may have a very strong heart, I may have a very brilliant brain, but I'm gonna have a miserable life because I'm in pain for 40 years. It would be enough to locally intervene on the joint, making sure that I can walk, making sure that I can hold my grandchildren, and so on and so forth. So that on its own, I think, has a value. But one by one, I think you can start really thinking about, okay, now that I have an aging heart, I can intervene on the heart. Now that I have an aging liver, I can intervene on the liver. Now that I have an aging brain, I can intervene on the brain. So I think that one by one, you could start potentially tackling all of those aging-associated disorders or indications. Now, I know the question is coming. To live forever? No, I couldn't care less about immortality. I want to make sure that actually we live as long as we are given, in a very healthy, happy, and meaningful way.
On that wonderful note, we will conclude this session. Thank you, Vittorio. This was absolutely fantastic and fascinating to listen to. Thanks to all our audience for tuning in, and until our next meeting. Have a wonderful day, everyone.
Thank you so much. Bye.